Good afternoon, everyone. I'm Janine Clayton, the Director of the Office of Research on Women's Health, and it's my pleasure to introduce our Wednesday afternoon lecture today. Dr. Lisa L. Barnes is professor in the Departments of Neurological Sciences and Behavioral Sciences at Rush University Medical Center. She's a cognitive neuropsychologist in the NIH-funded Rush Alzheimer's Disease Center. Dr. Barnes earned her bachelor's degree from Clark Atlanta University and her PhD from the University of Michigan, and she completed an NIMH postdoc at UC Davis before joining the faculty at Rush Medical College. Her research focus is on racial disparities in chronic diseases of aging, including Alzheimer's disease and HIV. She is the principal investigator of 3 community-based cohort studies of older African Americans and the director of the Rush Center of Excellence on Disparities in HIV and Aging. One of her cohort studies, the Minority Aging Research Study, known as MARS, is nationally recognized as a premier study of minority aging and has been continuously funded by the National Institute on Aging since 2004. Dr. Barnes has been recognized for her contributions to minority aging and health disparities by many professional organizations and has received numerous awards. She is the author of over 150 manuscripts, and she has also provided over 100 community presentations to older African Americans and their families. One of the defining moments of her career was the opportunity to work with HM, also known as the man with no memory, which represented a seminal case for neurological research in the 21st century. Please join me in welcoming Dr. Lisa Barnes. Her lecture today is Race, Risk, and Resilience, Understanding Racial Differences in Cognitive Aging and Brain Pathology.
Okay, thank you. Good afternoon. I would like to first thank Drs. Clayton and Bernard and the Women of Color Committee for nominating me for this prestigious lecture series. It is truly an honor to be here to present some of the work we've been doing in Chicago for close to 20 years now. I would also like to thank you in the audience and anyone watching on video for taking the time out of your busy schedule to come and listen to my work. I really appreciate the time. Before I start, I would like to just acknowledge the funding agencies for the work that we've been doing, as well as the study participants of the different cohort studies I will talk about today. Without these 2 sources of support, none of the work that we do would be possible. To give you sort of a layout of what I plan to discuss today with you in the next 45 minutes or so, I will review racial differences in cognitive function in the field in general and from some of our work. I will then talk about just select risk factors for cognitive decline and Alzheimer's disease in older African Americans. Then I would like to talk about some of the ongoing work looking at racial differences in underlying brain pathology. And hopefully these things taken together will help me sort of argue for the implications for understanding resilience in African Americans. And then I will end with gaps and opportunities and just talk briefly about some of the future directions that we're taking in our work in the very, very near future. So to contextualize the problem and make sure that we're all on the same page, I wanted to just present what the lay of the land is. It's well documented that African Americans perform more poorly on average than whites on cognitive function tests. This has been shown in numerous studies across the country for several decades. And here I'm just showing you 2 examples of the typical finding you see in the field when you compare African Americans and whites on cognitive function. And on this panel here is data looking at racial disparities in cognitive performance in mid and late adulthood from 2 very well-known studies, MIDAS and YCAP out of Columbia. So the panels on the top are data from YCAP, and it's showing you performance on 2 cognitive domains, episodic memory and executive function. And the panels on the bottom are the results from MIDAS. And what you see is that there are 2 lines in each graph, and the top graph in every case is performance of whites on these 2 cognitive function tests across different ages, and the lines on the bottom are the African Americans. So you can clearly see that across the ages, African Americans are performing more poorly than age-matched whites. Similarly, if you look at the data on the right, this is showing you the prevalence of cognitive impairment among Americans aged 55 and older from the Health and Retirement Study. And African Americans are shown in the blue bars. And you can see again that at every age, African Americans have a higher prevalence of cognitive impairment compared to Hispanics and whites. It's not advancing. Oh, there. This is also borne out by dementia incidence studies. Here I'm showing you data from Kaiser out of San Francisco, showing the incidence rates for dementia by age and ethnicity. And African Americans are shown in the purple bar, and as you can see, at every age after age 69, African Americans have a higher incidence of dementia than the other ethnic and racial groups that they measured. So this persistent disparity, coupled with the fact that the older African American population is rapidly growing, faster than the older white population, taken together, this suggests that the burden of cognitive impairment in Alzheimer's disease will be disproportionate for older African Americans. We don't know the cause of Alzheimer's disease, and currently there are no effective disease-modifying therapeutics. Given this stark reality, prevention becomes an important action area. And at Rush, we are squarely focused on prevention studies, where we identify risk factors for disease, both genetic and environmental, and we determine the biologic pathways linking risk factors to disease, so that we may develop strategies to prevent Alzheimer's disease and ultimately reduce disparities by either modifying lifestyle behaviors or identifying druggable targets and developing effective therapeutics. In order to do this, you need studies that include people without dementia, and you need diverse studies that include adults from the community beyond those who show up for medical care, because we know that minorities who have access to care differ on a variety of factors that could confound results in your study. You also need studies that include assessment of risk factors, regular testing to document incident disease, and blood donation for genetic testing if you're interested in identifying genetic risk factors. At Rush, we have several cohort studies that include these design features, and I'm only going to focus on 4 of them today, and I would just like to give you a brief description of the studies so that you understand where the data are coming from that I'm going to describe later in the presentation. So the first project is the Chicago Health and Aging Project, known as CHAP. It's a population-based study that took place starting in 1993. We did a complete census of all households on the south side of Chicago, indicated here in yellow. So here's Rush, just west of downtown, and CHAP is about 10 miles south of Rush. We did the census and invited all residents over the age of 65 to participate, and of about 7,800 who were eligible, 6,100 enrolled, and 60% were African American. All data collection occurs in person, in the home as interviews, in 3-year cycles, starting in 1993. In 2000, which would be the third wave, we began recruiting newly aged persons, people who turned 65. So the original and successive cohorts together yielded a sample size of 10,800 people. People come from very middle-class neighborhoods. as well as more affluent neighborhoods. And if you know anything about Chicago, it's very segregated by race. But these neighborhoods on the South Side are one of the few that have African Americans and whites living together in contiguous neighborhoods. So it really allows us to look at a range of socioeconomic status in both African Americans and whites. The other study that I will describe today is the Minority Aging Research Study, known as MARS, which began recruiting in August of 2004. This, unlike CHAP, is a volunteer cohort consisting of over 700 African Americans who are 65 years old at enrollment without known dementia, meaning that they don't have a diagnosis and they're not taking medications for dementia. They are recruited from churches, senior buildings, and organizations that cater to older African Americans. Testing occurs annually through in-home cognitive testing, risk factor assessment, and blood draws. Our follow-up rate has been very good, with over 90% follow-up among survivors. And for those, it's an ongoing enrollment process. So for those who enrolled in 2004, we have up to 13 years of follow-up if they're still living. And beginning in 2011, we started to recruit for autopsy, but it's still considered an optional component for people who choose to sign up for that part of the study. Now, MARS is completely African American, and it was designed to understand within race heterogeneity. But there are times when I want to understand racial differences, and so to do that, I need a comparison cohort. So for that, I need to tell you this first. Because it's community-based, we are very, very engaged with the community, and we use techniques that allow us to recruit a very enriched sample of older African Americans, or at least we try to. And our motto is, in the heart of the community, ever seeking to win the community's heart. And this is me here, and I found a group of people who had been getting together for 40 years to play tennis. And we took lunch, and I gave a presentation on healthy aging, and I was able to recruit about 75% of the people, and many of them are still in the study. So we do this throughout the community at different types of organizations to enhance our recruitment, and it's been very effective for not only recruitment, but for retention practices as well. Now, the comparison cohort that we use are two very well-known studies out of Rush, the Religious Order Study and the Rush Memory and Aging Project. These studies have been ongoing for 20 plus years, and they consist of over 3,000 older people without known dementia from across the USA, about 91% white. They all agree to annual detailed clinical evaluation with risk factors assessed and blood donation. And importantly, they all agree to organ donation at death. So that's a requirement to be in the study, something that you can never do in a minority population. But because they do this, they have over 1,200 autopsies in these studies. And importantly, there's a significant overlap in risk factors with these studies and MARS, and we use the same cognitive battery, allowing us to merge the data and look across race. All three studies follow the same longitudinal cohort design, where we enroll people at baseline with no disease, and then we document a range of risk factors. We follow them annually over time to measure change in cognitive function, and we're able to also measure incident disease in people who will develop disease eventually. And in people who come to autopsy, we get a range of pathology, from no pathology to more pathology, and we're able to correlate everything we learned about them when they were living to their trajectory of change over time, to the pathology underlying, to understand how the cognitive aging is playing a role in the aging brain. All in one design, one study. It's a really powerful way to look at aging in older adults. So just to remind you of the cohort studies, we have CHAP, which is the large population base, people living in contiguous neighborhoods, about 60% African American, with a range of SES across both groups. Then we have MARS coupled with MAP-ROS, volunteer cohorts. They're recruited from the community. They all enroll without dementia. They're followed annually, and they involve organ donation. Okay, so those are the cohorts I'll be talking about today. So I've already told you that cognitive function is is worse in African Americans across a number of different studies, and the prevalence of dementia is worse as well. But we know that cognitive function is influenced by many factors shown here on this diagram, and many of these factors also vary by race, making it very challenging to interpret black-white differences in cognitive performance when we see them. It also probably leads to an increased reports for cognitive impairment and risk of disease. And I'm going to show you some data that I think will illustrate that. Because our studies are set up to be longitudinal, we are able to look at change over time. And fundamentally, aging is a process of change over time. So if you're really interested in comparing blacks and whites, you have to be able to look at longitudinal trajectories. And when we do this in our studies, here's data from CHAP. I'm showing you on the y-axis is performance on a global measure of cognition. These are z-scores, so positive numbers are better performance, negative numbers is worse performance, and you have two lines. You have the whites and you have the blacks who started the study at different ages. And what you can see across the board is that you have these persistent level differences where whites are performing better than African Americans, but the lines are parallel. There is no difference in the rate of change over time when you look at people over periods of time. We have shown similar results in our volunteer cohort. Here I'm showing you data from semantic memory and perceptual speed tests. The whites are shown in the green bars on top, and the blacks are in the purple, blue bars on the bottom. Again, persistent level differences, but the length of the line shows you how long we followed people who started the study at different ages. You can see that the lines are parallel, and if anything, it looks like blacks are actually declining slower at older ages in these two particular domains. We've looked at this also with nonlinear change, because we know that if you follow people long enough, change stops being linear. You develop a nonlinear component. And when we looked at this in our cohort, our CHAP cohort, we divided the blacks and whites into two groups. Those who were alive throughout the follow-up period, shown in the line on the top, and those who died during the follow-up period. And what you see is that at about 6 years into the follow-up, you have a change point where a decline starts to go down precipitously, and this is the same point in both blacks and whites. And again, you see the level differences, but the lines are parallel. Now, we've also shown in our studies that much of cognitive decline seems to be driven by people who have Alzheimer's disease. So here I'm showing you data from European Americans, whites, and African Americans. The two lines on the top are people who never developed cognitive impairment throughout the study period, and the two lines on the bottom are people who develop Alzheimer's disease. And so whites are here on the top. They are performing better. Here are blacks who did not develop cognitive impairment. The lines are parallel again, but here in those who developed Alzheimer's disease, it looks like the African Americans actually are declining slower over time. Once, about 3 years before diagnosis, you start to see this sharp drop in cognition. And in fact, we have looked at African Americans with Alzheimer's disease and compared them to to whites with Alzheimer's disease, and we found that African Americans decline at about 25% slower rate than whites. So here's the line for whites with Alzheimer's, and here it is for African Americans. And you can see at some point there's a crossover. So to summarize these cognitive aging findings, we've shown that African Americans perform worse on average on tests of cognition and cross-sectional analyses, but there doesn't appear to be any evidence for faster rates of decline in longitudinal analyses. At least the data in our hands don't show any difference by race. But there is still this problem we have. We have this persistent level difference that we need to understand. And what we want to know is, can we identify factors that might predict decline? And what is the role of race? Because if we can do that, then maybe we can prevent decline or either slow the onset of decline. And importantly, are there factors that help us understand these persistent level differences? Now, we have looked at a number of different risk factors, some that are well established in the literature, and I'm not going to go through all of them, but I did want to present data on one genetic polymorphism that has been very controversial in the field when it comes to race differences, the apolipoprotein E4 allele. This is the most robust genetic risk factor for Alzheimer's disease in whites. But the frequency of the E4 allele is actually higher in African Americans, but the effect appears to be weaker. So in a number of studies, people either find that there is no relationship of E4 with Alzheimer's disease, or they find that the effect is much weaker. The disparity may be due to race-specific, I'm not doing anything, it's just going by itself. The disparity may be due to race-specific variation of haplotypes across ApoE and neighboring genes. We know from prior studies that the pattern of linkage between ApoE and a neighboring gene called TOM40 differs between African Americans and whites. In whites, the E4 allele is almost perfectly linked with TOM40 523 long allele. But in African Americans, the E4 is linked to 523 short in addition to 523 long. Could this explain the weaker effect of E4 on Alzheimer's disease in African Americans? Well, we looked at that in our data. And here I'm showing you data from our volunteer cohorts. The panel here on the left is the cumulative risk of Alzheimer's disease in whites who had the E4 and the TOM40 523 allele. And what you can see is that, it might be hard to see, but these are 6 lines, and the solid lines are the E4 alleles. So these are people who had or who were homozygous for E4, and these were people who were heterozygous for E4, and these were people who were non-E4 carriers. Superimposed are the dotted lines for the 523 allele, and there's complete linkage disequilibrium for 523 and E4 allele. You don't see the same pattern in African Americans shown on the right side. What you see here is the risk of E4, but the risk for the 523 alleles is not replicated in African Americans, shown in the bottom graph. We have similarly looked at the effect of E4 on cognitive decline in our cohorts, and we find that the E4 allele is associated with faster decline in episodic memory in both blacks and whites. So here are whites who have no E4 allele. In the dotted line are whites who had the E4 allele. You can see that it's a much faster rate of decline. The African Americans are the shorter lines because the study started later. And what you can see, though, is the same finding. These are the non-E4 carriers, and here are the E4 carriers, a faster rate of decline. But in contrast, when you look at another domain, like working memory, you see that the same effect in whites. The E4 is related to a faster rate of decline, but look at the lines for African Americans. They're completely superimposed. E4 is having no effect in this particular domain of cognition. We have examined a number of environmental and experiential factors in addition to the genetic risk factors. And there are many factors that increase your risk of disease, listed here on the slide, and many that decrease your risk. And one could argue that the ones that decrease your risk could really be resilience markers for cognitive aging because they protect you from disease, they slow the rate of decline. But interestingly, we have not found any of these factors to differ by race. We have found some for the increased risk that differ by race, and I'm not going to go through those today, but I think there's something that we can learn by looking at the decreased risk factors and trying to understand and model resilience factors. The question that we want to answer, though, is why do disparities in cognitive level exist? And to answer this question, we use data from CHAP again. Participants were randomly split into exploratory and confirmatory subgroups, and potential correlates of cognition were selected from a wide range of demographic, health-related, and experiential measures. In the exploratory subgroup, using a stepwise search algorithm, we examined the cognitive difference by race and then allowed candidate measures and race by candidate measures to enter the model. The cognitive score in the exploratory subgroup was a mean of 0.403 lower in blacks, and race accounted for 7% of cognitive variability. After the candidate selection process, 16 measures were retained. I'm not showing you the data for all 16, but in this model, which accounted for 45% of the variability in cognition, race was no longer significant when we had these two markers here, race by reading cognitive activity and race by neuroticism. The finding was the association of higher levels of reading cognitive activity with higher cognitive function was weaker in African Americans, and the neuroticism finding was that the association of higher neuroticism with a lower level of cognition was stronger in African Americans. But again, these candidate measures only accounted for 45% of the variability, suggesting that there may be other factors that we need to examine to understand cognitive aging in African Americans. And so in our studies, we are able to look at a number of race-relevant factors, because the problem has been typically in these large cohort studies, they focus on the traditional risk factors, but very few studies have incorporated race-relevant factors. And I know that NIA and NIMHD both have a research framework where they are really advancing for researchers to really consider a broader range of factors across many levels to try to understand health outcomes. So we've been doing this for a while in our studies, and I wanted to just present some of the data that we're seeing, and I'm going to just focus on the first three here in green. So self-reported experiences of discrimination is a very important psychosocial, a unique psychosocial stressor in African Americans, and it has been linked to a number of adverse health outcomes. And some, but not all, studies have found that it partially explains disparities in health. We were one of the first groups to look at the relationship of perceived discrimination to our cognitive outcomes in older adults. And what we found was that perceived discrimination was associated with worse episodic memory and perceptual speed in our participants, shown here in the red box. But much of the association was explained by depressive symptoms. We also looked at the effect of discrimination on C-reactive protein measured in the blood, and we found a very nice dose-response curve such that those who reported more discrimination had higher levels of CRP in the blood, suggesting a potential mechanism of inflammation linking discrimination to poor health. Another important risk marker in African Americans is perceived stress, which is the degree to which a person finds their lives unpredictable, uncontrollable, and overloading. We looked at this in the minority aging research study, and we found that persons who reported more perceived stress, shown in these blue lines here, had a faster rate of decline in both episodic memory and visual spatial ability. We asked a very simple question in our data set. We know where people were born and where they were living at age 12. And we wanted to see if early life residence would correlate with performance on our cognitive function tests. And what we found was that for African Americans who were born in the South or living in the South at age 12, shown in the red bars, these are Z-scores on our 19 battery cognitive function tests. Those blacks shown in red had poor cognitive function performance than the blacks born or living in the North at age 12. Now I was interested to see if I would see the same pattern in whites. And here I'm showing you the data for whites. It's in the same direction, but it's not nearly as dramatic as the difference between the blacks born in the South and blacks born in the North. And I just want to point out that we didn't have as many whites who were born in the South, which also points to the importance of understanding the social experience of older blacks in this country. We know about the migration patterns that happened from the South where people ended up in Chicago. And so performance on these tests is being reflected in where people started out. Now this could be dietary differences, health differences. It could be a host of differences. And we adjust it for a number of factors, and we still get the same finding. So this needs to be probed a little bit further to know what's going on here. We have just started with really preliminary data to try to understand the perceived discrimination finding a little bit further. And we've been doing functional imaging, functional MRI, and looking at how perceived discrimination is showing up in the brain. And compared to whites, African Americans with higher perceived discrimination showed greater connectivity between the insula and the left caudate. And we know from prior work that this network is important for emotional cognitive processing. So this perceived discrimination measure, it's real, it's doing something. We obviously need to have much higher numbers and to see if this will be replicated, but it's very compelling and interesting data to see how we can take a social experience and relate it to some underlying brain measure. So just to summarize the risk factors that I've just talked about, the effect of ApoE, which is a potent genetic risk factor, the effect of ApoE on risk of disease and cognitive decline in African Americans is likely driven by variation in the pattern of linkage with neighboring genes. We can identify risk factors that reduce the black-white difference in the level, but as I told you, that only accounted for 45% of the variability. So culturally relevant factors are important as well to understand the within-race heterogeneity and really need to be incorporated in these kinds of studies. But the question that I really want us to answer today is there something else going on? Race and pathology. We know from studies in whites that the accumulation of pathology starts decades before you actually start to see clinical symptoms. And in our studies in whites, where we have over 1,200 brains, we know that by age 85, Alzheimer's disease changes are present in nearly every brain, whether or not people showed, whether or not people had clinical Alzheimer's disease. We know that cerebral vascular disease is present in more than half of the brains, and Parkinson's disease changes are present in 40% of the brains, and up to 40% are positive for TDP43, which is a protein associated with frontotemporal dementia. And in fact, we've published that mixed brain pathologies, so a combination of all the common pathologies, is the most common cause of dementia in old age, followed by Alzheimer's disease pathology. But there's very limited data on neuropathology in African Americans, and none in African Americans who start a study without dementia. The data that are out there come from studies where people are at a memory clinic, they die and they come to autopsy, so they have the disease, or they're from autopsy series or medical examiners' cases where there's a little clinical information. So we want to know, is pathology the missing link in understanding the disparities in cognitive level and risk of AD? We know that African Americans have a high prevalence of vascular conditions like hypertension, diabetes. Could they potentially have more vascular disease in the brain? Now, we first looked at this in another cohort study that we have of people who were recruited from our memory clinic who had a diagnosis of dementia and came to autopsy, and we compared African Americans and whites. We found that mixed disease was the most common cause of dementia in both groups, but African Americans were much more likely to have mixed pathology as a cause of dementia, shown here with this red slice, compared to the little tiny red slice for whites. But one thing to point out is that all of these patients were from the clinic, and we know we discovered or examined what the mixed pathologies were, and we thought that they would have been Alzheimer's disease and infarcts, given the pattern of vascular disease in African Americans. But surprisingly, the mixed combination was Alzheimer's disease and Lewy bodies, which we didn't expect. Now we know that Lewy bodies are associated with behavioral problems, hallucinations, sleep disorders. So it's likely that African Americans who present to a memory clinic are showing up for behavioral problems and not for memory problems, which is why we're getting this combination of Lewy bodies and Alzheimer's disease pathology. But it has important implications for how we move forward as we try to understand what is the pathology that we're measuring in the brain and how are these therapeutics that we're targeting going to differentially work in different populations. And it also suggests that, or supports the fact that people who are just using clinic-based samples may not have a representative sample of the community, what's actually going on in the community. So that's the question that we are asking with our data, which is, of course, ongoing. You know, we're collecting brains over time, but we took an initial look at 202 deceased subjects who enrolled without dementia from our 3 volunteer cohort studies, the Minority Aging Research Study, the Memory and Aging Project, and the Religious Order Study. And we used consecutive deceased African American subjects with available pathology, and we matched them to deceased whites. We have over 1,200 whites, so we're able to match on key demographic variables that we know are related to cognition. And we quantified common pathologies using standard techniques. The neuropathologist was blinded to clinical characteristics of the participants. Here I'm showing you the characteristics of those 202 people. So we have 79 brains from African Americans matched to 123 whites. And you can see that they were matched on age at death, education, and the percentage that were female. The only difference between the two groups was that African Americans were much more likely to have the ApoE E4 allele at 42% compared to 19.8 in whites. And they were much more likely to have clinical Alzheimer's disease by the time they died, but it was not different from the rates for the white, 41.8 compared to 34.1. Remember, everyone starts the study without dementia, and some people develop it over time. Importantly, there were no racial differences in the burden of pathology. So unlike the clinic study that I just showed you, where there was more mixed pathology consisting of Alzheimer's and Lewy bodies, we saw no differences across race in these common pathologies that we measured. But we wanted to know, what is the impact of the pathology on risk of disease, and is there a differential impact by race? Here I'm showing you a Cox model where we enter the different pathologies to see what was related to risk of disease. And the values in red show you that both AD pathology and microinfarcts increased the risk of Alzheimer's disease, but there was a race by microinfarcts interaction, such that microinfarcts had a stronger association with clinical AD in African Americans than whites. And these models were adjusted for age, sex, education, and white race as the reference group. Now I want to show you what it looks like in a diagram. Here is when we just looked at AD pathology. So you can see that there's an increased risk of dementia the more pathology you have, but there was no difference by race. Then we added gross infarcts, nothing really going on there. But when we added microinfarcts, this is the race interaction that I just talked about. This is the effect in African Americans compared to the dotted line for whites. So a much stronger association of microinfarcts for African Americans than whites. So to summarize the pathology data, we've shown that clinic patients versus community-based participants without dementia at enrollment have different patterns of pathology. African Americans with dementia from a memory clinic are much more likely to have mixed pathology compared to whites, but there are no racial differences when you look at pathology from people who come from the community who started the study without dementia. The impact of pathology on risk of disease does seem to vary by race, however. Both AD pathology and microinfarcts each increase the risk of dementia, but the microinfarcts increase the odds more strongly in African Americans over and beyond the other pathologies. To conclude, the increased risk of Alzheimer's disease among African Americans that we hear about all the time is likely due to persistent racial differences in cognitive level. Culturally relevant variables need to be incorporated in studies of minority aging to understand the promoters of healthy aging. As I've shown you, we pointed out a number of risk markers: perceived discrimination, perceived stress, early life residence, reflecting stressful social conditions or disadvantage. These were associated with decline. suggesting that modifying or intervening on the ways that people experience stress may be targets to promote resilience. While the burden of pathology does not appear to differ, the effect on AD risk was different. Unexpectedly, we did not see more vascular pathology despite the well-documented burden of vascular risk. But are there other vascular pathologies that weren't measured, that maybe we haven't even discovered yet? Or are there different regions, like the watershed areas, that may be playing a role that we need to look at further? I think this points out a number of gaps and opportunities that we can incorporate to move past disparities and improve health equity for our older African Americans. And it's just a few key points, but I think they're important. Number one, we need to increase minority populations in clinical research in general and in these aging studies in particular, because as we've shown, the drivers of disease may be different. We also need longitudinal designs if we're going to compare African Americans and whites, so that we may distinguish factors that affect test performance from true change over time. We need studies with biologic outcomes, studies that include genomics, proteomics, metabolomics, et cetera, because we currently have a limited understanding of biologic mechanisms underlying disparities. And we need to understand why there are level differences. It's a problem that we have these persistent level differences that are leading to more impairment, and we need to start looking at prevalent conditions like the adverse social conditions, but also taking a complex systems approach that takes into account the interaction of risk and resilience factors and across the life course. Because one thing that we've learned is that these disparities that we're seeing are occurring when people start to study at age 65. So they're not just happening after 65, they're in place already, suggesting that we need to move the lens back earlier and be able to look at what's happening in midlife, so where we have more of an opportunity to actually make a difference and maybe change the trajectory of what's going on in old age. There are exciting biomarker and drug discovery projects. Rush is participating in some of these AMP-AD projects, the studies from whites, but there are very few minorities enrolled in these initiatives, and it's because you need brain tissue. But the area of science that's moving forward the fastest is not including the minority population where the risk seems to be greatest. We need to do something about that to change that scenario. So just to briefly touch on the future directions that we are taking in these studies, we are likely to be doing more neuroimaging to understand the neurobiologic substrates of cognitive aging in people while they're alive. We also are funded to look at decision making, to understand the determinants and adverse consequences of racial differences in decision making. We know very little about that. Could that be a window to understanding disparities? And we have a new investigator at Rush who just received a diversity supplement to conduct a mixed methods approach to study barriers and facilitators of brain donation in underrepresented populations, including African Americans, Latinos, and low SES whites, so that we can start to improve this picture of limited data from these underrepresented groups. Now, this work is not done by one person, obviously. We have a fantastic center at Rush, multidisciplinary, with people doing all kinds of really fascinating work and lots of exciting new things going on. And I'd just like to acknowledge the many, many co-investigators who have participated in some aspect of the work that I presented today. Thank you very much for your attention. Oh, yes, I can take questions if there are any.
Did you look at the differences between Black women or African American women and men?
Our aging studies are about 70% women, because women live longer. So we don't really have the power to do that. I can try to look at it, but I'm not sure I would find anything. I really struggle with getting African American men enrolled in these studies. I've tried all kinds of techniques. I just got, I got a recommendation today to look at the VA to get more men. But that's a really important area that we want to look at in the future.
Mm-hmm. Thanks. If I can have two quick ones. One was, are the microinfarcts more prevalent in your African-American participants than in the white participants?
We did not
just
see
stronger risk?
We did not see more microinfarcts in the African-Americans.
And the second, could you elaborate on your idea that you tossed out about watersheds? Do you have in mind something to pursue on that?
We know that a lot of vascular disease is occurring in the watershed areas. We currently don't look at the watershed areas in our studies. We look at about 8 different regions in the brain to measure both gross infarcts and microinfarcts. So that's just an area that's missing that we think maybe, because we expect to see something when it comes, there's no reason that with all of the hypertension, diabetes, that you're not seeing this in the brain. So we just want to look at the watershed areas to see. We don't really have a specific hypothesis around it. It's just missing from our protocol currently.
You mentioned about some imaging methods, including MRI, but we have great techniques to measure both the amyloid plaques as well as the tau using PET imaging.
Yes.
So that is a much more predictable biomarker for diagnosis. Obviously, there is no therapy yet. And also, the second question is regarding the use of aspirin. Aspirin has some effect on some of these issues. Did you look at some of these victims or patients, whether there is some intervention in the early stage so that will affect the microinfarct and other pathology infarct-related from
right, so
platelet thrombosis? For
your first question about PET versus MRI, you're exactly right that PET would be a much better way for us to look at it. But these are community-based participants who we would have a challenging time recruiting them for PET studies, where we're going to inject something invasively into their veins that we don't know what the effect's going to be later. Just for purposes of keeping the community's trust, I would like to have a bit firmer idea about what PET is doing over the long term before I introduce it. Now, we have a clinical core in our Alzheimer's Disease Center, and we have been talking about incorporating PET into that. That's also all African Americans. We have a Latino core. So that will be coming. But I haven't yet gotten the courage to do it in my minority aging research study. I don't want to lose people. It's already hard enough getting the brains. So for your second question about the aspirin, we have not looked at that, but we do a visual inspection of all medications every year. And we have medications coded and entered into our database. So that is something that we can look at to see if it's affecting any of these factors that I talked about today.
Just another note, for diabetes, at least the racial factors have been much more predominant, identifying different genes affected in diabetes.
Right, we looked at diabetes and it was associated with worse performance, but we didn't see a race effect. It was equally detrimental in blacks and whites.
Thank you. Good luck.
Thank you.
Thank you very much for that superb presentation. I couldn't help being struck by the potential link between perceived discrimination, activation of the renin-angiotensin system, as we see quite a bit of that in cardiology, as well to the link with the microvascular disease. And I wonder if you're studying the activation of the renin-angiotensin system as a mechanism for the cognitive loss, or the differences in cognitive function between race.
That's an excellent suggestion. We're not looking at that, but that is an important mechanism that we should incorporate. Thank you for my next grant.
Actually, I may have your next grant after that. Amazing, amazing talk. Thank you so much. And you may have actually addressed it, but you said that there's a Latino core, because Latinos, particularly Mexican Americans, are also at very high risk for Alzheimer's disease. And many of the variables that you talk about are, dominant in Latinos as well. I mean, go through any old nursing home in Texas, and you're going to find the exact same thing, and hallucinations and things along those lines. Except it's not going to be, the signs are like no Mexicans and dogs allowed. as opposed to having been raised in the South. So how far advanced do you have? It sounds like you've got the white and the black group, and it doesn't seem like it would be that expensive to have the Latino group.
Actually, we were just funded 2 years ago to start a Latino cohort in our Alzheimer's Disease Center, funded by NIA, and the PI of that is Dave Marquez, and he is a kinesiologist out of UIC, and we have enrolled about 170 Mexican Americans from Chicago who are getting the exact same cognitive battery, of course, translated and back translated in Spanish, and measuring many of the same risk factors. So we will have some of that information, but the study is brand new and they are very young. We have decreased the enrollment age to about 55. So it'll be really interesting to see if we get change in this 5-year period.
And also, Chicago is different than the Southwest
Yeah.
when it comes to discrimination. But it's amazing that you've been able to fund that.
Thank you.
Hi, I was just wondering if you've taken this internationally and looked at African, blacks in Africa, basically, and the effect of maybe America lifestyle.
I haven't done that, but there is a study out there by Hugh Hendry that compared African Americans in Indianapolis to Nigerians in Africa, and he sees very different risks of disease. The Africans have a lower risk. There's many differences. Obviously, the African Americans in Indianapolis are more sedentary, the U.S. diet, all kinds of things. He also sees differences by APOE. So I would love to be able to do an international study. I'm trying to get this one perfected,
Yeah.
and so maybe that will be down the line.
Great, thanks.
Thank you.
Hi, Lisa.
Hi.
Great talk, thanks.
Thank you.
I'm an avid tennis player, so I want to hear about the differences between tennis players and non-tennis players. But aside from that, you talk about the mean differences between the African Americans and whites, and we know there's a lot of variability in both groups. I'm curious if you found within each group there being high performers that sort of differ from the norm, and whether there are differences between the high performers, whatever the cognitive tasks might be, between the racial groups.
Yeah, that's a great question. I have a slide that I didn't show where we took a subsample of about 200 blacks and whites, and there's clear heterogeneity in cognitive decline, with some declining precipitously, some staying stable, and some actually improving. So there is a lot of heterogeneity across both racial groups. We haven't looked at risk factors that might predict that yet, but that's something that we could do in the future. Tennis, right? Oh, one thing that I should mention is that I love to share, and we have data available. You can go to our website at Rush, and you can get any of our cohort studies. We'll send you a data set, we'll send you brain tissue, we'll send you bloods, and you can create the next great study of minority aging. So please consider looking at our website and seeing what we have available. We have lots of wonderful cohorts, and we're very open to sharing.
So please join us in the library for a reception that is sponsored by FAAS. Thank you, everybody.